Good afternoon, everyone. I'm Rick Barron, the Executive Vice Chancellor for Health Affairs at MU and the Executive Director of the NextGen Precision Health Initiative. Thank you for attending the 10th session of our NextGen Precision Health Discovery Series. These discussions are intended to highlight critical precision health topics and approaches. We have asked our speakers to share their knowledge in ways that engage all of us, and we hope that they do so in a way that sparks new ideas, new collaboration, potential, and new areas for next-gen research. As you listen to these wonderful talks, we would like you to all think about research opportunities in precision health, where you can contribute and where you can help identify potential collaborations across the University of Missouri system. Today, it's my pleasure to introduce our speaker, Dr. Hemachandra Reddy. Dr. Reddy joins us from Texas Tech Lubbock University Health Science Center in Lubbock, where he serves as a tenured professor and vice chair of basic and translational science. He is also a professor in the departments of neuroscience, pharmacology, neurology, public health, and speech and language and hearing at Texas Tech. Dr. Reddy's lab has been involved with research on healthy aging, dementia, other neurodegenerative and metabolic disorders. Additionally, Dr. Reddy is a prolific writer, authoring more than 200 peer-reviewed articles, book chapters, reviews, and he serves as the Chief Scientific Officer for a Lubbock-based small business company called AB Synap Techs, LLC. I asked Dr. Reddy today to focus on Alzheimer's disease and the research demonstrating how physical inactivity and unhealthy diet contribute to the disease process. He will discuss potential impact of healthy lifestyles and delaying Alzheimer's disease progression. And with that, I will turn it over to Dr. Reddy. Welcome, my friend Hemi.
Thank you very much, Rick. It's a great honor and share some of our laboratory findings on healthy aging and healthy lifestyle and reduced risk of dementia. So I sincerely apologize if I say any unclear passages, and I will be happy to answer, address after my presentation at any time. My contact details are there. Let me open the screen. Now this time, very good. So yeah, these are contact details of my, people can email me or contact me via LinkedIn or Facebook, whatever way it works. So it's, okay, this is the brand new building opened recently for administrative building. You can see behind the research labs on your left-hand side, a physician's pavilion where patients get into the building, and is kind of admin building in the front, research labs, and the health UMC, they're well connected. So a faculty, particularly by internal medicine, they are here and then they go back to see patients. It's very convenient. Of course it is, we recently celebrated 50 years of our health science center. A lot more things are happening around. And I came to Health Science Center 7 years ago. That was, 2014 October, 7 years. It's a very productive and very, excellent experience dealing with students, postdocs, and faculty colleagues, running around all the time, writing grants and sharing interactions and whatnot. So the next slide is going to be, this is the slide when I visited, the University of Missouri, Columbia, a NextGen Precision Health building. You see my long-term friend Russ in the middle, me the other side, Rick, and Rick showed us the building we really enjoyed, and I really wanted to show you this picture. Okay, so this is the photo of my lab members taken a year ago. Several new members are missing, and it's an amazing experience. I sincerely thank all my lab members for their kind support and doing things what I say. I listen to them most of the time. What Dr. Reddy, we want this, we want that. I'm 100% with them. So it's being a wonderful journey for me. And you see the two guys here, Rodney McWilliams, my right hand, understands me, supports me, taking care of a lot of things for me. On the other side, Jeremy Garcia, he is the purchase man. We need certain items. Second day, they will be there in the lab, anything, everything. So he's, without them, it's very difficult for me to run the lab. Of course, I would like to acknowledge, thank funding agencies who supported my research for last 2 decades, starting with NIH, several R1 grants, Alzheimer's Association. When I did not have any grant, those are the people stood by me, gave me the multiple grants, and then from there, journey started. And as Rick mentioned earlier, we do have a small business, a Lubbock-based business, Epson Optic LLC. We recently received STTR grant for our discoveries of DDQ molecule to delay aging and dementia in elderly individuals. Garrison Family Foundation supported me for first 5 years of my journey here. I was a executive director for Garrison Institute, immense support. Still their family foundation still supporting me, my wife, across the campus, TTU. And I also received a large amount of support from CH Foundation, local foundation, multiple grants. And in 2015, we received a TTU, TTHSC Presidential Collaborative Grant from here. And remaining below, Medical Research Foundation of Oregon, Alzheimer's Disease Center, Oregon Health and Science University, Oregon Partners for Alzheimer's Research. And those are the things I received when I was at Oregon Health and Science University. 15 years of journey, wonderful, wonderful, wonderful. And when I was a fellow with Francis Collins and Danilo Tagle, I received $50,000 grant from Huntington's Disease Society of America. It is, they allowed me to take that money when I went to Oregon Health, and also received large amounts of money from several pharmaceutical companies. Some I listed here: Vertex, Medivation, and Colobius. Okay, my lab research has been divided into 3 units. The first unit is focusing on molecular basis of aging and Alzheimer's and other neurological diseases, and with the focus on synapses, and mitochondria. That's what we have been doing science for the last 20 years. Second unit is mainly focused on drug discovery to delay the aging and Alzheimer's disease. We use quite a bit of cell cultures and animal models. Of course, we received recently, STTR grant from NIH. And this coming January, we are applying for phase 2 clinical trials. Our application for IND in the process. So it's a lot of excitement here. And we also have early detectable biomarker development unit. So we are using a small amount of blood, isolating, serum and plasma, looking at RNA-seq based microRNAs. It's going very well. My former postdoc got K99, and now he's a junior faculty. We are trying to recruit him as a tenure track. Things are going very well. So in addition to that, we also involved with diabetes, obesity, kidney disease, cancer, cardiovascular, and nutritional. More recently, for the last 2 years or so, healthy lifestyle and COVID. I'm glad to say a new book is coming out a few months from now with the 20 chapters focusing on healthy lifestyle, COVID-19, and dementia. Okay, so I'm very active in running all sorts of stuff here. I listed a few here. So we run Alzheimer, aging and Alzheimer's disease seminar series. You can click on it. You can find recent thing. Next week, I will be giving another seminar, 20 years of Alzheimer's experience, dedication to patients and the families. We do have an active aging and Alzheimer journal club going on. And we have a public health. I'm being part of a public health faculty, so I run the journal club there. Our lab encourage, entertain women in science. If you click on that, you'll see so much going on. Almost 50% of them are women. and we encourage them, we nurture them and pushing them. So it's an amazing experience. Of course, like as Rick mentioned, we published over 200 articles. And I know Russ may be tired of reviewing my articles from the other side. Thank you very much, Russ. So this is the overview of my today's presentation. I'll talk about the global impact of dementia. I'll give you some background information about Alzheimer's disease and Alzheimer's dementia. Of course, I'll talk about cellular and pathological changes. And I'll show you some data which is relevant for today's presentation. And the concepts of today's presentation is healthy aging and better lifestyle, diet and exercise. And I'll show you some summarized tables based on the mouse models and human elderly individuals, MCI subjects, and Alzheimer's patients, of course, summary and conclusions. Okay, this slide shows the global impact of dementia. As you can see, 1 in every 3 seconds, a new case of dementia is being diagnosed. And you look at the numbers, according to 2000 estimates, 50 million people globally, including 6 million Americans, suffer from IAD. This number is expected to go up to 82 by 2030 and 152 million by 2050. If you look at the total estimated worldwide healthcare cost, 1 trillion based on numbers in 2018, that is expected to go up to 2 trillion by 2030. It is a huge problem in the society, dementia. So this slide, let me minimize this up so that I can see. Very good, thank you. So prevalence of Alzheimer's disease from 1900s to 2050. So what exactly this slide is telling us? We started living longer. The longevity is going up and the dementia is following. So we are not escape from the dementia when we reach 85 and beyond. Almost 50% of the world population are going to be demented if we do not do something significant in terms of healthy brain aging. The green bar is kind of slow and steady, progressive, but that indicates age between 65 to 74, and the blues markedly increased over 1960s to expected up to 2050, and the most significant one is red bar, 85 and beyond. You see so much. That means we started living longer, longevity is increasing, the problem of dementia is also increasing. Okay, this slide is showing the dementia rates in high income and low and middle income countries. In high income countries, the numbers are pretty stable, 2015 to 2050. Whereas the low and middle income countries, numbers are estimated to increase markedly. It's maybe because of lack of good infrastructure, diagnostic facilities, and also socioeconomic conditions. So on the other hand, high income with all the good infrastructure and diagnostic facilities, most important awareness about dementia. So that's what it is stable. Now, this slide shows the guy who discovered a disease is a German physician, Alois Alzheimer's. So they named after him. That's what you see, Alzheimer's disease, Alzheimer's dementia, several. So just want to clarify 1 or 2 things. When I go and give seminars, the clinicians and researchers get confused. What is dementia? What is disease? So that's what I just put for a lay audience here. Dementia is the clinical term, come of for memory loss and other intellectual abilities serious enough to interfere daily life. That's the dementia. On the other hand, a disease is a pathological one when the brain is damaged with morphological pathological changes, that leads to dementia. According to CDC, Alzheimer's disease is a common cause of dementia, affecting 50 to 70% of all dementia cases. Of course, Alzheimer's dementia is categorized into three groups: mild, early Alzheimer's, moderate, middle stage Alzheimer's, of course, severe. So people with MCI, that is mild cognitive impairment subjects, have a likelihood of developing Alzheimer's over the period of time if they don't take proper lifestyle conditions and care. Now you see here on your left-hand side, the intact brain with white matter, gray matter is the whole no neuronal loss. On the other hand, right-hand side, severe neuronal loss. You see mainly holes, no white matter, even tiny bit of gray matter. That is Alzheimer's disease. So that happens, in people with Alzheimer's. Now, I don't know how I can take this stuff out. Okay, God, it's gone. Okay, so I put three different definitions of aging. Let us start with what is aging. Physiological changes occur in all systems. Examples: cardiac output decreases, blood pressure increases, and the lean body mass declines with age. Middle one, let's see. As we age, brain naturally changes, affecting the memory, learning, loss, and also shrinks with age, and changes occur in the vasculature. Of course, aging is a major risk factor for multiple age-related conditions, including Alzheimer's, Parkinson's, and cancer, and others. What is healthy aging? Oh, one more. Adopting healthy habits, behaviors, staying involved in the community, using preventive services, managing health conditions, understanding all the medications that contribute to a productive and meaningful life. And the other definition, see if I can get it out, is, okay. So the process of developing and maintaining the functional ability that enables well-being in older age, that is according to WHO definition. So brief introduction about Alzheimer's disease. It's a progressive neurodegenerative condition characterized by the memory loss and multiple cognitive impairments. Currently, over 50 million people worldwide, including 6 million people, suffer from Alzheimer's. 2/3 of women, 1/3 of men are at lifetime risk for Alzheimer's. Sometimes people say it is a woman's disease. It's not really. It's both men and women are affected, but more in women. Maybe they live longer. Brain body physiology are different. So many things we need to understand. Major pathological hallmarks, everybody knows: amyloid deposits and intracellular neurofibrillary tangles. But these are not the ones correlate with the cognitive decline. Synapses, loss of synapses, synaptic damage are the best correlates of cognitive decline. And loss of synapses and mitochondrial damage are early events in the process, in the disease process. What causes Alzheimer's? Only we know 1 to 2% total AD cases, with genetic mutations in 3 different loci: amyloid precursor protein, presenilin 1, presenilin 2. Causal factors are still unknown for vast majority of the AD cases. Of course, Down syndrome cases with the trisomy 21 exhibited amyloid pathology and cognitive impairments and stuff. Other factors are apolipoprotein E4, is affecting the major risk factor, not only in dementia and other chronic conditions, hypertension, others as well. Lifestyle activities are the major ones, including unhealthy diet, physical inactivity, and occupational environmental exposures. Type 2 diabetes, stroke, and traumatic brain injury and other conditions also contribute to late onset Alzheimer's. Above all, aging is the number one risk factor. If we live up to 85 and 90, we are going to be demented. It's a matter of how much, 10%, 20%, 50% are total dementia. This slide shows the risk factors I covered pretty much earlier. On the left-hand side, non-modifiable. We can do very little or nothing. Age, sex, genetics, genetic polymorphisms, and mutations. On the other hand, we can do so much about these factors. Modifiable, it is in our control. Eat healthy diet, do exercise every day. We can delay, progression of the disease, remove radicals from our brain and body, and other conditions also, diabetes, obesity, metabolic syndrome, smoking, depression, and cognitive reserve and traumatic brain injury. So this slide shows the major cellular changes in the disease progression. Several years of intense research globally, implicate that multiple cellular changes are involved: mitochondrial fragmentation, loss of synapses, amyloid deposition, glial and astrocytic activation, and neurofibrillary tangle formation. All these changes leading to neuronal loss and dementia. In addition to these, cell cycle regulation, hormonal imbalance, and many others. Based on these changes, therapeutic strategies have been developed, currently being tested in animal models and AD patients. I will say there is no drug or agent that can delay the disease process. In June, FDA approved monoclonal antibody based on amyloid beta called aducanumab, if I say correctly, approved conditionally. This drug is highly debated in the community, and it's not a good stuff. Of course, I don't want to say anything bad or anything. Time will tell the future of this drug. I can only say there is a nice review article published in Nature earlier that will tell the pros and cons of this drug and other things. So this is my favorite slide for students of amyloid beta precursor processing, how amyloid beta can form in the neuron. So there are two pathways. One is the non-amyloidogenic pathway. It is based on alpha secretase and here, and then the other one is beta secretase pathway. In non-amyloidogenic pathway, the amyloid beta domain is cleaved right in the middle, leaving soluble APP alpha and C83 fragment. And amyloidogenic pathway cleavage occurs right at the beginning of the A beta domain, and also gamma secretase here. So this leaves 40 and 42. 40 is a smaller form of amyloid beta fragment, and 42 is a longer one, more toxic one, participate in the oligomeric formation and photofibrils. These fragments accumulate in subcellular organelles such as mitochondria, ER, endoplasmic reticulum, cause subcellular dysfunction that happens in Alzheimer's. On the other hand, in healthy subjects, both beta secretase and gamma secretase leaves small fragments of beta domain. They clear from the brain all the time. On your right-hand side here, you see the demonstration of amyloid beta and full-length APP using our mouse model with 6E10 antibody. This is, you see this toxic peptide, 4 kilodalton. Now, this slide shows the amyloid beta deposits in 12-month-old APP mice in the top panel, 24-month-old APP mice on the bottom panel. You see the difference. What is the difference? Aging, 12 months and 24. Same mutation mice carry, but you see in 24 months, they're all over. And 12 months, kind of mainly on hippocampus. But what happens in familial, sporadic cases, oxidative stress, mitochondrial dysfunction induce A beta production. This slide shows the inflammatory response based on glia and astrocytic activation. We tested using the two different markers of microglia and one GFAP. You see, amyloid deposits are surrounded by tiny glial cells. Their job is to engulf, follow the amyloid deposits, but they couldn't. It's insoluble and it's very difficult to handle. They don't give up. They produce more glia. That leads to inflammation in the brain. And this is my favorite slide, axonal transport. So here is the soma, and here is the terminals, synapse, right? Postsynaptic, presynaptic terminal, postsynaptic terminals. This is the area they need more energy, more cargos, more vesicles. What happens in a healthy neuron, the tau, my cytoskeletal protein tau tightly binds to the microtubule, the black here, allows cargos to move from soma to the terminals nicely. What happens in Alzheimer's neurons? Tau becomes phosphorylated and mostly detached from the microtubules. It cannot bind. That allows the cargos and my other organelles not to move to the terminals. That leads to synaptic starvation degeneration. Now, mitochondria, as I mentioned earlier, synapse and mitochondria are my favorite things I study. So mitochondria perform several, important functions in the cell. They produce ATP energy. They maintain intracellular calcium, and also they produce free radicals. And at the same time, they also sites of free radical scavenging. So. And if my cell is about to die, they push it through and clear from the, through the apoptotic mechanism. So they're also responsible for the activation of caspase family of proteases. Okay, we need to go much faster. Okay, so mitochondria are synthesized in the cell, in the center of the cell here. They travel all along axons and dendrites and produce ATP for several synaptic functions: vesicle fusion, neurotransmitter release, neurite outgrowth, synapse formation. These processes are highly energy dependent. And mitochondria move from soma to the terminals through kinesin-based enterograde manner. And then they go back and to recycle through dynein-based retrograde manner. So they they live only 2 to 3 weeks. So basically they synthesize new mitochondria and they recycle to maintain the energy in this, in the neuron. So I would like to show you some data to justify my today's presentation. When I got a job at Oregon Health and Science University 2001, so I wanted to, I did my postdoctoral experience, with Francis Collins and Daniel Ottaglia on Huntington's disease, another neurological genetic disease. Alzheimer's is kind of sporadic. It's very, if you don't understand the aging process, you don't understand the Alzheimer's. So that's the reason I found a model called TG2576, overexpressed 2 mutations, 670, 671 frame shift. So I found the mouse, we housed these animals for a long period of time. You can see and looked at the mRNA changes in the cortex and hippocampus, 3 different stages. Stage 1, 2 months, 5 months, and 18 months old. We did compare the mRNA levels with the wild type, age-matched wild type. And we analyzed the gene expression data using clustering analysis, northern blot, and in situ hybridization. And in one cluster, we found all mitochondrial encoded genes. So that's the curiosity. I was expecting to see more synaptic problems. On the other hand, we found mitochondrial genes upregulation. You can see here, pretty much they're all mitochondrial and encoded genes. Here, lane 1, lane 2, lane 3, wild type mice, 2 months, 5 months, 18 months. Lane 4, lane 5, lane 6, 2 months, 5 months, 18 months, Alzheimer's mouse. You see the gradual increase of mRNA levels in Alzheimer's mice. That's one thing we found. So maybe due to compensate the defects of mitochondria. That's what our interpretation at that time. Not only APP mice, but also we see gradual increase of mRNA levels of, in this case, NADH, yes, it's complex 2, complex 1, in the wild type. What does it mean? Maybe aging process also involved in addition to APP. Then we tested what is the link between mutant APP or soluble A beta and mitochondria. So what we did, the very primitive experiment we did the immunostaining analysis using the 8-hydroxyguanosine antibody as a marker for DNA damage, oxidative DNA damage, and we see increased immunoreactivity in the hippocampus and cortical layers in the APP mice relative to wild type mice. So indicating that oxidative DNA damage involved in the disease process. So that was our gene expression thing. We proposed this model. We hypothesize that mutant APP may be soluble A beta, even oligomeric A beta enter mitochondria, induce free radicals, and lead to mitochondrial dysfunction damage. So to compensate that mitochondrial dysfunction, the mitochondrial encoded ones upregulated. That upregulation sustains in young animals, middle-aged animals, but not in aged animals. So that leads to decompensation, neurodegeneration, and memory loss. This was our model at the time, 2002. And then what we went on and testing our model. What is the link between mitochondria and amyloid beta? So this slide shows the purity of mitochondria isolated from the cortex and hippocampus. Then we run those mitochondrial lysates. You can see the TG2576 wild type. We found 4 kilodalton amyloid beta in the APP mice, but nothing in the wild type. So then this is I showed you earlier for a shorter form of A beta and longer form of A beta. That's what we did in the NextGen. We ran those protein lysates, mitochondrial protein lysates on a gradient gel, 4 to 20%. We could see a doublet, 40 and 42. That means both the forms are associated with the mitochondria. Then next experiment, what we did measure a soluble A beta 40 and soluble A beta 42, and also hydrogen peroxide. That's one of the things, with the free radicals. So the 2 months and 17 months, they positively correlated with soluble A beta 40 and 42 with the hydrogen peroxide, meaning free radicals are produced in the mitochondria. The next experiment, what we did, former postdoc Marcus Calkins, he was the expert in primary cultures. Very first time, I would say, he killed the day 1 pups and cultured the hippocampal neurons and cortical neurons. And we looked at several aspects. I'm just showing you because of time thing here. Electron microscopy of our APP cultures here, and then the wild type. You don't even see the mitochondria, dead impression of mitochondria. They're fragmented, increase in number, so many here, and very small here in the wild type. So the fragmented mitochondria increased, and mitochondrial length is decreased. That means there is a problem with mitochondrial morphology and intactness. So the next experiment, what we did, we wanted to see dendritic spines. These are the things, communicate the neuronal, from neuron to the next neuron. And we measured the number of dendrites and dendritic length in APP mice, 12 months, that those are symptomatic. They show cognitive problems at that time. So you see significantly reduced dendritic spines in the APP mice. We also looked at the tau mice, another pathological hallmark. We see the similar pattern, reduced dendritic spines in the tau mice as well. So whatever I showed you so far, just checking my time. So why we have to study antioxidants and exercise and stuff? We did see free radicals production. We did see reduced dendritic spines, mitochondrial DNA damage, and synaptic ATP is reduced, and age-dependent increased mitochondrial damage. That's important. And also we see cognitive decline in mice linked to mitochondria and oxidative stress. As I mentioned earlier, the other things, physical and mental inactivity, unhealthy diets and habits. So these are the things rationale why people started looking at treating mice and people with antioxidant supplements and exercise and stuff. So, so some of these antioxidants I listed below tested in mouse models of Alzheimer's, of course, elderly individuals and Alzheimer's patients. Some I listed below here: vitamin C, vitamin E combination, and ginkgo biloba, melatonin, curcumin, the Indian spice, coenzyme Q10, lipoic acid, and many others. And our lab and many labs are also working on mitochondria targeted molecules. As I mentioned, mitochondria produce free radicals. If you introduce, administer some of these molecules with antioxidant properties, they can scavenge free radicals in the mitochondria. That's what we have been working on, multiple molecules, MitoQ, SS31, DDQ, our own lab discovery, and other molecules as well. Next few slides, I will show you the summary of studies done on animal models of Alzheimer's disease with multiple antioxidants. You can see several, in Alzheimer's field, you can see over 50 animal models made. TG2576 is the first one made by Karen Shaw, and then the tau, and then the, ApoE4, another risk factor for Alzheimer's, and double mutant mice, APP-PS1, and NSC APP, and some people made knock-ins of APP-PS1, and of course, triple mice and 5X FAD, and these several supplements and in the diet, they tested on these mouse models, and these are very long time. I mean, starting from 1 month to up to 8 months in mouse, right? So they found very beneficial effects in animals fed or treated with these supplements. Out of 21 out of 23 antioxidant mouse studies have shown beneficial effects. That means these antioxidants are in the diet and supplement working very well in animal models. Next slide shows in the elderly individuals. You see this several studies, different geographical areas in the globe, and they antioxidant and in vitamin E, vitamin C combination. These are longitudinal studies. You can see anywhere from 2 years to up to 30 years. So that data is very authentic. You have to believe it. So 9 out of 12 studies, antioxidant studies, have shown beneficial effects in elderly individuals. Now, Alzheimer's patients, randomized clinical trials did not do well with antioxidant supplements, vitamin E, vitamin C combination, because if you look at Alzheimer's patients, the damage is already done. A small number of neurons left in the brain. And so if you give these treatments, you are only helping astrocytes and glia because there are no neurons left. So that may be one interpretation. I may be totally wrong, but that's what these clinical trials, antioxidants did not do well. Now, fish intake. I want to show you this, my favorite. I eat a lot of salmon and other fish. So they keep the brain intact, hippocampal volume and stuff like that. I'll show you some data, on the people with the who ate or intake of fish and incidence of cognitive outcomes. So you can see several studies. Pretty much almost all studies did show beneficial effects. People see these are the starting with 3.9. That is a short fish intake study, and up to 21 years. They all showed beneficial effects. So, it's good to have more fish. Okay, next. This is my favorite stuff, Mediterranean diet. I pretty much follow the same diet every day. So eating, what is this Mediterranean diet? Primarily plant-based products, fruits and vegetables with grains and legumes and to some extent nuts and stuff like that, and replace the butter and with a with healthy fats like olive oil and things like that. And eating fish is also good stuff. And of course, red wine with the certain and other things are there. So no sugars, no processed meat. That's the bottom line. And here is I would like to show this pyramid with all these fruits and vegetables at the bottom of the pyramid, and the fish and seafood, good stuff. Poultry is good, but no meats and no processed meats and no sugars. So that's a very tiny amount. I know we cannot control someone's lifestyle, someone's diet and stuff, but if you reduce those and develop eating plant products and other things, it's good stuff. Now, so coming to the Mediterranean diet summary table here, and people with number of studies, several studies here from different parts of the globe, number of subjects, large number. You look at that one, the bottom one is 6. Wow, good God, 16,000 non-demented. It's like 6 years. These are very longitudinal studies. Pretty much almost all studies showed reduced incidence of Alzheimer's or MCI. That means the Mediterranean diet is good for health. Of course, 2 studies did not see much significance, but otherwise, almost all studies showed reduced dementia or MCI in with more high MMSE mini-mental score examination numbers. Now, the next few slides I will show you a physical exercise. Believe me, I live close to next door to Higginbotham Park. That one circle I make, that park is a 0.73 miles. I make every day 3-4 rounds. So I just walk. So I want to summarize some of these things: physical exercise and link it to reduced, amyloid pathology, increased cognitive function in mouse models of Alzheimer's disease, individuals mild cognitive impairment, and of course Alzheimer's patients. Some examples of physical exercise: walking, gardening, water aerobics, swimming, yoga, meditation. I do quite a bit. Of course, Tai Chi. I saw at Oregon, a neuroscience head was doing Tai Chi every day and bringing all the people, his lab people and the colleagues, to Tai Chi. And of course, laughing is good physical exercise. And this slide shows the physical exercise and the cognitive behavior in Alzheimer's mouse models. And you see so many different lines of mice. Pretty much most of them are, they're mixed up for tau as well. And most of them are amyloid beta mice. So these animals put on a treadmill, and then they looked at their cognitive behavior and amyloid pathology and tau pathology, the reduced amyloid beta and tau pathology, and of course improved cognitive behavior. So almost all studies have shown beneficial effects of physical exercise on Alzheimer's mouse models. Now, the same thing with subjects with mild cognitive impairment. So the MCI is a stage. Here is healthy status. Here is MCI, and here is the early Alzheimer's. So if you are at MCI stage, if you take proper care, meaning healthy diet, regular exercise for a period of time, and you go back to a healthy status. If you don't do those things, you move to the early Alzheimer's. That's what exactly so many scientific studies. Currently, my lab is conducting one study, 4,000 individuals, healthy, they say healthy, 60 to 90. We are looking at their brain scans, looking at their blood profile. Of course, MoCA, Montreal Cognitive Assessment Test, is good for the healthy subjects. and also their blood profile. On the other hand, why some people live without any cognitive problems. So that's what we are trying to understand: their genetics, their lifestyle, their exercise habits, whatnot. On the other hand, 500 Alzheimer's patients. So we are looking at their brain scans, of course, MOCA test assessment, and the blood profile, and why and how. So we're trying to understand some individuals live longer without cognitive impairments or things. So the point I'm trying to make, mild cognitive impaired subjects, they are right in the middle. If they do good job, they go back to normal brain health. If they don't take care of themselves, they go to progressive, cognitive impairment. So this study is pretty much same thing. What I'm trying to show you is the physical exercise on humans with mild cognitive impairment. So pretty much improved after doing the physical exercise. And these are quite a number of studies, except one study. Pretty much all studies show physical exercise good for cognition in MCI subjects. Even in Alzheimer's, patients as well. I mean, if you do pretty much everyday exercise, so you can bring back your memories and do good stuff. That is the summary of this particular table. If somebody is interested, I can talk about it in detail. And this is my summary table. That is we have another 14 minutes to discuss. So what exactly is the mitochondrial impairment and synaptic damage are key factors for aging and Alzheimer's, I mentioned earlier. So diet, antioxidant intake in supplements and diet have shown beneficial effects in Alzheimer's mouse models and elderly individuals and mild cognitive impaired subjects. But antioxidants intake did not do well in Alzheimer's patients. I have to say that. So another point I want to make it here, fish intake actually reduces the risk of MCI and Alzheimer's and dementia. Mediterranean diet reduced the risk of AD and improved cognitive functions in elderly individuals and MCI subjects. And physical exercise is good for all groups. Elderly individuals, MCI and early Alzheimer's, even younger population, younger individuals, like, if you do good exercise, your memory is sharp, you can do good stuff in the schools and colleges. So, the physical exercise, I stress that it is very important for all age groups. Of course, I end up saying that healthy diets, regular exercise, and better lifestyle will delay the progression of dementia. Not only dementia and other, chronic conditions such as hypertension, kidney disease, and respiratory illnesses. It's healthy diet is and regular exercise is good for diabetes, obesity, and other conditions as well. So I will stop it here, and I will be happy to answer. There are I see some chat things here.
Thank you, Hemi. That was great. And Mary, do we have some questions?
Yes, we do. We have a few that have come in. Dr. Reddy, the first one I have here is, what were your main methodologies during this research?
Methodology. So the first part of the laboratory research, I can tell the techniques and stuff. And the second part is I did the thorough survey on mouse models of physical exercise and antioxidant intake. This is the data collected from PubMed search and the healthy diet and exercise and clear. And the first part of thing is very clear. I mean, laboratory methods like immunoblotting analysis, immunofluorescence analysis, and electron microscopy, and Golgi Cox staining, which you see the dendrites, number of dendrites and dendritic spines that determines the cognitive assessment in mouse and humans. That's what we did. I mean, it's varieties of techniques, transmission electron microscopy, the state-of-art technique, which measures the number of mitochondria and length, whether they are intact or broken with the crystal and stuff like that. That's what we did. That is for the last 20 years we have been doing. And in our grant, all the techniques are involved. I mean, that's the way we put in cell culture stuff, mouse models, and postmortem brains, and looking at the similar thing in different ways. And we have a grant. We're trying to get for $4 million grant on lifestyle in mouse. That's interesting. So 2 lines of mice we put together, and some exercise and diet, or aim 1 and aim 2, and combination together, and you'll see how that goes. That's the the lifestyle is the key for Alzheimer's, not only for Alzheimer's and even other chronic conditions. You can pour millions and millions of dollars on drug discovery. I work on the same thing, but outcome is very not so good, because we know if we take care of ourselves from birth to elderly, even early, late, and we are good, instead of eating unhealthy diets and exercise. Absolutely. I do not know how much I could answer that.
Well, thank you, and good luck on that next submission that you just talked about. This next question is from Dr. Sahota. Excellent presentation, Dr. Reddy. Sleep and cognition is a fast developing area. Can you comment on the role of sleep or sleep deprivation on mitochondrial function?
Absolutely, that is my favorite subject. If you look at the Alzheimer's literature, or even other chronic conditions literature, we need sleep. We actually tested in the lab, though we did not publish yet. So we keep animals awake for a while, and we measure following day the blood and amyloid beta levels. So if animals are not sleeping well, they do produce more amyloid pathology, more kind of cognitive problems in mice, at least our own laboratory experience. Same thing goes with other chronic conditions. Enough sleep is important so that the brain can rest, and they clear all the free radicals in the neurons, and then go from there. If we do not sleep, of course, in humans, so that leads to following day screwing up, and eventually developing the early cognitive problems in humans. I would say animal models, yes, people tested. David Holtzman, next door to you guys there, St. Louis, his focus is mainly on sleeping and dementia, sleeping and other chronic conditions. It's outstanding. He's done thorough job. Who knows if I happen to come.
Mary, why don't you get to some of the other questions, or some good ones?
Thank you. And then there's a question of what is the age of old mice? What is the age of the older mice that you used in your research?
Okay, that is important. So I have to tell you that we recently published a paper, came out in Redox Biology. My former postdoc, he made a mouse model for microRNA 4553P, the transgenic, and also he made a knockout of the same microRNA. The transgenic microRNA expressing several copies of 4553P, those animals lived 6 months longer than the wild type. So beautiful survival data. So generally in wild type mice, they live up to 24 months, I agree. But in Alzheimer's mice, they die prematurely, 15 months to 20, maximum. But these animals, the microRNA mouse model, transgenic mice, they live up to 40 months. So, it's amazing. So it all depends on how animals they produce, how much free radicals they produce in the brain and the rest of the body, and how they, sort of cope up with the conditions. Absolutely. So
Thank you.
No free radicals, they live longer.
Is there a study going on in Columbia at NextGen?
Well, Dr. Reddy is not in Columbia NextGen.
Right.
So Dr. Aileen Lin just joined us, and she's also on this. I see her viewing. She just arrived from Kentucky, and her goal is to get some study started in the NextGen building.
Absolutely, absolutely.
So this next question is, if someone follows all of the diet, exercise, et cetera, recommended and gets Alzheimer's disease, what is your thinking now regarding why?
You mean after following a healthy diet and regular exercise, still they develop Alzheimer's?
Yes.
So those individuals, I can say that maybe they carry genetic mutations. I mean, the Alzheimer's is pretty much 99% of them are sporadic late onset, right? Only 1 or 2% are familial cases with early onset. If you take care of all your healthy diet and exercise, still you develop cognitive problems, means you have a genetic factor there that is keep producing the amyloid beta or tau and all the toxic elements in your brain. That may be the case, but in the vast majority of cases of all the late onset, right? So what I mentioned, healthy diet and exercise takes care of so much. Yeah, I think genetic component is the is the one I can say in those individuals.
I think that really leads into the next question here, which is, does ancestry affect the risk of Alzheimer's disease? And you kind of just spoke on that.
Yeah, I mean, the familial cases, right? Familial, some individuals carry the mutations and polymorphisms, so we can still take care of as much as possible, reduce the toxins in the brain and body. Certain things we can do. But for example, Huntington's, right? They carry genetic CAG repeats, and then the midlife disease for 5-10 years they die, and there is no cure for to eliminate the toxic effects of expanded polyglutamine repeats. We kind of say same story. If you look at the presenilin 2, very early onset, people with presenilin 2, they develop cognitive impairments when they are 25-30 years. Can you see? I mean, people always say that Alzheimer's is late onset. Yes, it is 99% of the cases, but 1 to 2%, early onset. I mean, they produce more amyloid deposits, more tau phosphorylation, and all bad things happens very early on in life with the genetic mutations. Presenilin 2, even presenilin 1, also very toxic mutation.
Going to get the last two.
This last two, yeah. Would taking omega-3 DHA be a viable exchange for increasing fish intake?
Yeah. I'm not expert on that, but the answer is yes. So yeah, absolutely. That helps.
And then this last question: any evidence that exercise improves mitochondrial function? Could be any other mechanism?
Absolutely. There is a grant. I didn't want to tell all the stories. If you do the exercise, autophagy and the mitophagy, kind of exercise is, activate mitophagy. What is mitophagy? Clearing the dead mitochondria or dying mitochondria. So exercise helps activate mitophagy, even autophagy thing. So you do exercise, you measure your RNA-seq data and your blood. Of course, you cannot do things on the people brain because they're alive, right? But mouse models, yes, you do exercise, you put them on the treadmill for 6 months and kill them, collect the blood and collect the brain tissues and do autophagy, mitophagy markers, you get the answer. Believe me, the exercise, you activate the mitophagy, meaning clearing the dead mitochondria. Neurons are clear from all the toxins.
Thank you, Dr. Reddy, for sharing this presentation on healthy aging and reducing the risk of neurodegenerative diseases by lifestyle alterations. It was just an excellent look into the impact of this progressive disease and the factors that could lead to it. Please join us again next month for our NextGen Discovery Series, when Dr. Giovanni Guidoboni will present Engineering Precision Health. Dr. Guidoboni is an Associate Dean of Research at the University of Missouri's College of Engineering and a professor with joint appointments in electrical engineering and computer science and mathematics. Her research focus is on mathematical modeling and data science applied to engineering and life sciences. Her presentation will start at noon on Tuesday, December 7th.
December 7th, yeah.
So everyone, and Dr. Reddy, you're welcome to join that as well. So, everyone have a great Thanksgiving holiday, and we'll see you back after Thanksgiving on December 7th. Thank you all very much.